Good evening, everyone. Am I audible? Are you able to hear me?
Yes, ma'am.
Yeah, thank you. So let's begin with our week 5 session of the online NPTEL course, Drug Delivery Principles and Application. Let me just share my screen. Can you see the presentation in a slideshow mode? Yeah, okay. So have you guys gone through the week 5 lecture material? So that it becomes easier for us to understand the questions and subsequently answer them. Anybody has gone through it?
Yes, ma'am.
Okay, so I'll just give a quick recap. So we have been talking about particles, most probably nanoparticles and microparticles in the last session. And then we saw how they are uptaken and how they're eliminated, what all things play a role in their activity, such as shape, size, charge. And then we also saw that what are the optimum parameters that a particle during drug delivery should have so that you maximize its effect for an effective therapy. Next, now so we have seen few concepts of drug delivery. The session is now going to move from drug delivery to tissue engineering. And before moving to the actual tissue engineering, we'll be discussing something about protein adsorption. So that is the theme of this week 5 lectures. So proteins, as you all must be knowing, are polymers of amino acids, and they have both hydrophobic and hydrophilic parts. And because of this property, because of being having both hydrophobic and hydrophilic parts, they are able to adsorb on the surfaces. Now, why does this become important? Because whenever, let's say, during tissue engineering or any therapy, you're injecting a material. Let's say you're putting a knee replacement, and you have an implant inside the patient body. So these proteins are going to adsorb on that implant, and that's how they're going to modulate the cellular response as well. So that's why it becomes important for us to know how proteins are going to adsorb on the implant surface and how can we avoid them or promote them depending upon our application. So whenever, there is it happens sequentially whenever you have a biomaterial, let's say even if you have a polymer nanoparticle or you have a steel implant and you have injected that into a patient body. So first of all, it's going to be in contact with a lot of water, and then in that water, in the body fluids, you'll be having a lot of proteins which are dissolved, which are soluble. And so first of all, the implant is going to react with the water, and then the proteins present in the body fluids are going to adsorb on the surface, and now they are going to dictate what cells are going to adsorb on this implant. So first you have a water, then you have a protein layer, and that protein layer is going to modulate your cellular response. Now this becomes important whenever we talk about immune response, because we don't want the graft which you have injected, if which you have implanted, or any biomaterial which you have implanted, to have a very severe immune response. If that happens, then that will lead to graft rejection, and on top of not treating the disease, you will again introduce some other defects which will be further difficult to treat. So we don't want that. Also, sometimes we cell attachment becomes an important uh, point for us, because whenever, let's say, again in the case of knee replacement, you have injected your implant in the knee and you want it to be integrated with the patient's body. So at that, in this particular example, you want the cells and you want the proteins to adsorb onto that implant. So depending upon the what application you have for your biomaterial, the strategy to manufacture these biomaterials changes. For example, if you don't want any immune response, you will manufacture your implant, your biomaterial, in such a way that its surface will have less absorption of proteins. On the other hand, if you have implanted an artificial kidney or an artificial liver, you want that organ to be a part of the patient's body. In that case, you will want the cells to interact, you want the proteins to interact with this graft. So this is all the session talks about. And so from moving from protein absorption, some part of tissue engineering is also introduced in the last two lectures. If you guys have gone through it, you must be knowing. So how can we define tissue engineering? So tissue engineering is a combination of cells, materials, and engineering principles along with some growth factors. And why do we do this? We do this because we want to improve a particular function of an organ, or we want to replace a biological tissue in the patient's body. So you must be knowing a lot of examples of tissue engineering. For example, you have artificial skin grafts to treat burn injuries. You have contact lenses in case you want to avoid specs. So, and there are various ways of tissue engineering, various methods in which a tissue can be grafted. First of it is that we can generate entire tissue. Let's say I want to replace a heart. We can generate an entire heart in vitro and then implant it into the patient's body. Or you can generate a particular part of a damaged organ. Let's say half of your liver is damaged, and you want to only replace half of that function. So you can partly make this liver in vitro and then implant it into the patient's body. Or there is something known as decellularization of scaffolds. So you take let's say there's a donor and you want to implant this particular liver into a patient's body, but you don't want response towards that donated liver. So what you can do, you can decellularize this liver. You can
Ma'am,
hello.
ma'am, are you changing with your slides
No,
or
no,
your
no, no, I'm
slides?
not. I have not yet begun with the assignment. I'm just
Okay,
giving
ma'am.
a quick recap of the whatever lectures have been taught in this week.
Okay, ma'am.
Yeah, thank you for the concern. Yeah, so I was talking about decellularization. So let's say you have a donated liver. What you can do is you can decellularize it using surfactants. You can remove all the cells and just keep the ECM matrix, and then you can implant this into the patient's body and allow the patient's body to recellularize this ECM. So in this way, it will have lesser immune response compared to the first case. So that were the concepts covered up in this week, and now we can move towards the questions. Does anybody have any doubt before we move on towards the questions? If not, we'll begin. So the first question, can you guys see the now are the slides changing? You can see the questions, right?
Yes, ma'am.
Yeah.
Yes, ma'am.
So the first question is, why do proteins absorb on material surfaces? Pick one option. You have to select one correct option. I'll give you guys some time to read through the options and let me know if you guys have the answer. We just discussed it, so if anyone has missed it, you can give it a try.
Option 1.
Okay, we have one answer. Option one. Thank you for the response. Anybody else? Any other answers? Okay, let's move on. So you were correct. The correct answer is first option because they're weak surfactants. So proteins, as I said, have both the parts, hydrophobic as well as hydrophilic. Let's see what are surfactants first. So this is how a surfactant looks like. It has, so these are known as amphiphilic molecules. Molecules or compounds which have both hydrophobic and hydrophilic parts are known as amphiphilic. So so since your proteins are also surfactants, but compared to your actual detergents, they are very weak surfactants, and they have both of these hydrophilic and hydrophobic part. So whenever any surface, so a surface can be hydrophilic or hydrophobic. So whenever proteins come in contact with the surface, they can easily change their shape and their conformation to adapt to the surface. For example, if you have an hydrophilic surface and proteins that are present in a body are already in a hydrophilic environment. So they can easily attach onto this hydrophilic surface. On the other hand, if you introduce a hydrophobic surface, a protein which has its hydrophobic parts buried inside it, it can open up, it can bury the hydrophilic parts now, and its hydrophobic parts can be exposed out, and then that can attach onto the surface. So that's why do proteins absorb onto the surface? Is because they're weak surfactants. They can easily modulate their shape. Is it clear? Any doubts? Shall I go ahead? Okay. So the next question is, which of the following two statements are false about protein absorption in biomaterials? So we have to select two false options, and there are several options listed here. You guys can go through it one by one. For your benefit, I'll just read through it. Limited absorption sites and consequent competition means that not all proteins in the plasma phase can be represented on the surface. Driving forces for absorption are intrinsic surface activity and bulk phase concentration of the proteins. Once a protein gets absorbed, it doesn't come off. Surfaces vary in selectivity of adsorption. Adsorption is highly dynamic. Proteins always lose their structure and function after adsorption. Biological activity of the adsorbed protein varies on different surfaces. So we have to choose false statements. Anybody wants to volunteer? I'll give you guys a few minutes to think over it. Anyone?
Option 3 and 6.
Option 3 is once the protein gets absorbed, it doesn't come off. Okay, 3, 4, 5, 6 proteins always lose their structure. Okay, we have one response. Thank you for answering. Anybody else? Okay, let's look at the answer. So both of your options were correct, whoever answered. So the right answer is once a protein gets absorbed, it doesn't come off, and proteins always lose their structural function after absorption. Let's see why both of these statements are false. So there's something known as competitive adsorption happening whenever a material is introduced, whenever proteins wants to absorb nanomaterial. So let's read through the slide. The equilibrium adsorbed protein layer composition determined by total and relative protein concentrations. It's a very complex and poorly understood phenomenon. So which protein is going to adsorb more on the surface is determined by their relative concentrations. Let's say protein A has a lot of concentration, so naturally that is going to adsorb more onto the surface, and this is a very dynamic process. So it's not like once a protein is absorbed, it will forever stay there. No, it can easily detach depending on if there is a more competitively absorbing protein in its vicinity, it will just displace that protein and absorb onto the surface. So whatever this option says, once a protein gets absorbed, it doesn't come off is false because it can easily come off. And the next is proteins always lose their structure and function after absorption, which is also false because so you guys might be knowing about protein denaturation. We say a protein is denatured when it loses its shape, its conformation, and consequently its function. So protein denaturation upon absorption on a surface is also a variable phenomenon. It's not like all proteins will denature to the same extent. It will depend upon what material is there on the surface. So whenever it says proteins always lose their structure and function after absorption, which is not the case. There are some proteins which are very, very stable in the body, and even if you introduce any material, they do not denature to this extent. So that's why both of these options are false. Everything else listed out in the options is the correct information about protein absorption in biomaterials. Is it clear?
Yes, ma'am.
Thank you. So I'll move on towards the third question. Consider that cells are resuspended in a rich protein solution, say FPS. This suspension is added on top of a biomaterial. Rank the interaction of different components, cells, water, and proteins with chronology of their interaction with surface of biomaterial. So we have to rank whenever a biomaterial is introduced. We need to rank what is going to interact with the biomaterial first. Let me know if you guys have the answer.
Option
Third
3, ma'am.
option.
I guess there were two responses. Can you answer one by one?
Option 3.
Option 3. Okay. Anybody else trying
Water,
to?
protein, and cells.
Water, protein, and cells. Okay, we also have the same option 3. Okay, thank you both for answering. Anybody else? Okay, no other response. Let's check the answer. Yeah, both of you were correct. The right option is option 3. So whenever a biomaterial comes in contact with the body, first the water is going to interact. If you guys have gone through the lecture material, you must have seen this slide. So the sequence of events when a material is placed in a biological environment, first it interacts with water, which is also known as wetting of a material. Then it interacts with the proteins, and after the proteins, it interacts with the cells. Any doubts in this? Okay, so people who are like responding in the messages, I won't be able to see the screen once I'm in the slideshow mode. So it's okay if you want to answer, you can just unmute your mic and let me know the answer. Or if you guys have any doubts, you can stop me at a particular point and we can discuss further. Okay, then I'll move on towards the next question. The question is, what are the common techniques to quantify protein adsorption on material surfaces? We have to choose two correct options. Any guesses?
First and last option.
First and last, that is Western blotting and surface plasmon resonance. Okay. Any other? Thank you for answering.
Yes, madam. First and last.
First and last. Okay. Thank you.
Ma'am, sixth and last.
Okay.
Ma'am, sixth and last.
Sixth is 1, 2, 3, 4, 5. AFM. Okay. AFM and surface plasmon resonance. Okay. Thank you. Any other response?
Ma'am, first is confirm, but another is confused.
Okay. Sure. Sure. First. Okay. Let's see if we. Let's look at the answers. So the right answers are Western blotting and surface plasmon resonance. So whoever answered first, you were right with one option, and the other was surface plasmon resonance. So these are the methods where we can quantify protein adsorption. First is we can, let's say you have a biomaterial, you put it in some serum or body fluid extracted from a patient, and then you incubate the material with it, and then you centrifuge it, and then you subject it to something known as SDS page. Another method is Western blotting, which is followed with SDS page. Once you have a page gel run, you will see the bands separated. You can transform them to a membrane, and then with the known antibodies, you can just get a profile. That's where you can detect which protein is there adsorbed on the surface. And then there is surface plasmon resonance, which looks something like this. So whenever you have anything attaching to this surface, this angle of the reflection from this light source changes. So based on that, you can quantify. First of all, you can identify what protein it is, and then you can quantify it. So whoever answered these two was correct. Whoever answered AFM, AFM can be used to trace out the surface of a material, but here we have to quantify it. So AFM can just give you an image of what is there on the material surface, but it is not capable of quantifying how much protein is there. So I hope I was clear. So the right answers are Western blotting and surface plasmon resonance, since we want to quantify the protein absorption. Is it clear? Okay.
Yes, ma'am.
Yeah. Thank you. I'll move on. So the fifth question is, what is Roman effect? And we have to pick one correct answer. Just read through the options slowly. Let me know if you guys have the answer. If not, we'll discuss.
Second option.
Second is
Option B.
okay. Both of you answered second.
Second option, ma'am.
Okay.
Second option, madam.
Okay. All of you are answering second option. I'll just read that. Replacement of kinetically favored proteins by thermodynamically favored proteins from a surface over time. Okay. Anybody else wants to volunteer? Thank you for answering. I'll wait for others to read through it. Okay, let's move on. Let's look at the answer. So all of you were correct. The correct option is replacement of kinetically favored proteins by thermodynamically favored proteins from a surface over time. This effect is known as Roman effect. So as we saw in the previous question as well, there is something known as competitive adsorption. So whenever you have a biomaterial exposed to proteins, initially the kinetically favored proteins are going to adsorb on that surface. Kinetically favored means whichever proteins are in the vicinity of that material and whichever is in high concentration is quickly going to adhere to that surface. And that's why first of all you will have a layer of these kinetically favored proteins, but as time goes on, other proteins which are thermodynamically favored, which means that attachment of these proteins is thermodynamically favorable. It is more stable. So these kinetically favorable proteins might not have a stable interaction with this biomaterial. So whenever these stable interaction proteins, which are also known as thermodynamically stable proteins, come into the play, they can easily displace the previous proteins and they can now form a second layer. So this effect is known as Roman effect. Is this clear? I hope I'm making sense to you people. Any doubts?
No, ma'am.
Okay, thank you. Then I'll move on. So the sixth question is, when nanoparticles are injected in vivo, the proteins form a layer on the surface of the particles. What is this layer called? Nucleophilic substitution of proteins, nucleophilic addition of proteins, protein corona,
Ma'am,
electrophilic.
third option.
Option 3, ma'am.
Okay, thank you.
Option.
Third option, madam.
Okay, all of you are saying option 3. Anybody else has any other answer? No? Okay. Yes. All of you are correct. The correct answer is protein corona. So this is what happens when you inject a nanoparticle inside the body. The proteins are going to form layers like this. So it can be a hard corona or a soft corona based on how stable that layer is. So proteins which are very, very highly stable on this adsorbed surface, they will first form a layer and it's very difficult to displace them. Now, depending on what proteins are here, other proteins might interact with them and form a secondary layer, which is known as a soft corona. So that's all the question asked is. Next is, if cells are transplanted from a human donor to a human acceptor, what kind of transplant is it? Allogenic, autogenic, xenogenic, or antigenic? Yes?
Ma'am,
Option
first option.
Okay, both of you, I'm assuming, is answering the first, that is allogenic. Okay. Anybody else? Thank you for answering. Okay, let's see. Yeah, all of you are correct. The right answer is
I'm
allogenic.
first option.
Oh, sorry, I didn't wait for your response, but thank you for responding. Yeah, so the correct answer is allogenic. So allogenic transplant is referred to any transplant that is taken from the individual of the same species. Let's say from a human to human or from a pig to pig. Both of these transfers will be called as allogenic. Against that, what is autogenic? Autogenic is taken from the same individual. So let's say there's a patient and we want to treat a particular disease. So we take some stem cells from him, process these stem cells and send it back to the same patient. Let's say we do some gene therapy with the stem cells and then implant it again into the same patient. That will be known as autogenic. What is xenogenic? Xenogen is taken is a concept. I mean, xenogenic transplant is something taken from individual of different species. Let's say we have a patient and the patient immediately needs a liver transplant, but we don't have a let's say we don't have human liver available and we have a pig liver. So as I said, we can process some part of it. We can decellularize that, recellularize it with human cells, and then put it back into the patient. So xenogenic is taken from individuals of different species. Is this making sense? Did you guys get this? Antigenic is something totally different from transplants. It comes in case of immune response. Any material or particle which is capable of generating an immune response in the body is known as antigen. Are you guys getting this?
Yes, ma'am.
Okay, thank you. Shall I move on? Okay, the next question is for transplantation in humans, allografting is one of the most common approach. What are the major drawbacks of this approach? We have to select two correct options. The major drawbacks of allografting, that is transplantation in humans.
Ma'am, first
Option one
and
and two.
second.
First and
One
second.
by one, please. I
One
guess
and two,
so.
ma'am. Option.
First and second.
Okay. All of you answered first and second. Okay. Thank you. Anybody else? Others can also volunteer. I think it's the same people answering if I'm going correct by the voice. I can't see your names. Sorry for that.
First and second option.
First and second. Okay. Thank you. Let's see. So all of you were correct. The correct options are lack of donors and spread of disease. So whenever we transplant something, so allografting is taking a graft from one human and implanting it into another human. So although this is the most widely used practice right now in clinics because of lack of other practices, so there are certain drawbacks associated with this transplant. First is lack of donors. We so there are a lot of demand is there for organs, let's say eyes or heart, but we don't have enough donors, so that is one drawback. Next, there is spread of disease. For example, if you transplanted liver from one patient to another, and let's say that liver transplant had to be done very immediately, so there can be a possibility that not a thorough check was done with whatever was if there was a problem with the donated liver, and you just transplanted over the period of time, maybe not immediately, after a while you can see that if that liver was already damaged, if it had any kind of disease, any kind of pathogen inside it, that will be now spread to our new patient, and that will be a problem. So that is one drawback. And another drawback is immune rejection, where since even if it is from the same species, it is still a foreign organ for our body. So whenever you implant an organ from one human to another, it can lead to immune rejection, and this reduces the half life of organs. Is this clear? So because of this, we need alternative options such as tissue engineering. That is just to emphasize the problems that are currently faced by people. Shall I move on?
Yes, ma'am.
Okay. The next question is, which surface is more favorable for protein adsorption and why? We have to pick two correct options.
Option 2 and 4.
Option 2 and 4.
Ma'am, first and last.
Okay,
No,
so
option 2 and 4.
okay, so I have two responses that say option 2 and 4. I have one response saying 1 and 4. Anybody else? Anybody else has any other combination of the options? Thank you all for answering. We'll discuss. No other response? Okay, let's see. So the correct answer is option 2, that is hydrophobic surface, because it is energetically more favorable, and option 4, that is rough surface. So whoever answered 2, option was correct. Whoever answered 1 and 4, fourth option was correct. So let's see. So as I said, your proteins are going to have both parts, hydrophobic as well as hydrophilic. So since these proteins are already present in body, they are in contact with a lot of hydrophilic. Since they're in contact with water, they have their hydrophilic parts exposed out. Now, if you put in a surface, so these proteins are also going to have some hydrophobic part exposed out, because not all hydrophobic part gets buried. So whenever you put a surface, if it is hydrophobic in nature, they are very happy. They don't want to, the proteins don't want to interact with water, so they can easily adsorb onto this hydrophobic surface because it is more favorable for them. So they can just displace the water and attach onto the surface. The other option, so that's why hydrophobic surface is more favorable for protein adsorption. On the other hand, whenever you have a rough surface, because uh, let's compare. If you have a smooth surface versus a rough surface, smooth surface is going to be very smooth. It will not have any indentions or it will not have any rough patches. On the other hand, if you have this rough surface, it might have spikes over it. So this leads to more surface area. So that's why more the surface area, more is your protein absorption. So whenever you have a material and you want proteins to absorb on it, you should have a hydrophobic surface because it's more favorable, and a rough surface because it has more surface area. Are you guys getting this? Any doubts? Okay. So let's move on towards the last question of the session. So this is a numerical. Consider that the proteins adsorbed on the surface of an implant using a monolayer model according to Langmuir adsorption isotherm. This is the formula where theta is the ratio of sites occupied by the molecule versus the total sites available for adsorption. K is 2 per millimolar and P is the concentration of the molecule. A cytokine is trapped inside a chamber at a concentration of 2 millimolar. If there are 10 to 5 total binding sites, how many of them will be occupied at steady state? We have to assume equilibrium conditions and we have to write our answer in a multiple of 10 to 4. So your answer will be just one numerical digit. So you guys want to give this a try? It's a straightforward formula. You just have to put in values and I'll give you guys some time to solve through this. If not, we'll go over the solution step by step.
Ma'am, 8.
Okay, we have
8, ma'am.
answer as 8. Anybody else? Thank you for answering. Others can take some more time and solve this. Okay, let's look at the answer. Yeah, whoever answered was correct. 8 is the right answer. So we'll just go over the solution. Seems pretty easy. So you have this formula given where theta is sites occupied by total sites, and we have this expression. we have the value of K with us and P with us. We just put it here. And we also know the total binding site. So let's first solve for theta. So once you put all the values in the formula, you get this, and the answer is 0.8. And now we have a formula for theta, which is sites occupied versus total sites. We also know the value of total sites, and once you solve this, you'll get x as x. I'm denoting x as the sites occupied as 0.8 into 10 raised to 5. Since the question is asking us to answer in a multiple of 10 raised to 4, you just convert this. So the right answer will be 8. So whenever you are answering such questions in the exam, if it's already mentioned that it's 10 raised to multiple of 10 raised to 4, you just put in the first numerical value and not the entire expression. Everybody got this? Does anybody have doubts in any of the questions? For people who joined late, I'll just go over the questions quickly. Any particular question you want me to discuss? If not, I'll just quickly go over the questions once again for people who have joined late. So this assignment started with this question: why do proteins adsorb on material surfaces? It's because they are weak surfactants, because they have both hydrophobic and hydrophilic parts, and it's easier for them to modulate their shape whenever a surface is introduced. So that's why proteins adsorb, because they are weak surfactants. Next question was, which of the following two statements are false about protein adsorption in biomaterials? And the correct answer was, once a protein gets adsorbed, it doesn't come off, which is false, because protein adsorption on the surface is very dynamic. Whenever a protein gets adsorbed onto a surface, any other protein which has more stable interaction with the surface can displace this protein. So this option is false. Next option is, proteins always lose their structure and function after adsorption, which is also not correct, because depending upon the surface, depending upon the protein, if there are several stable proteins, very strong proteins, which once adsorbed, there's no need for them to denature. So the denaturation of proteins is not a fixed phenomenon. The next question was, consider that cells are resuspended in a rich protein solution. This suspension is added on top of a biomaterial. If you rank the can somebody, whoever has unmuted the mic, can you please mute your mic? Thank you. Yeah, so we were going over this question. So we have to rank the interaction of different components with chronology, and the right answer was water, protein, and cells. So material first comes in contact with water. So the right answer was water, protein, and cells. Next question was, what are the common techniques to quantify protein adsorption on material surfaces? And the right answer was Western blotting and surface plasmon resonance. Next question was, what is Roman effect? And pick one answer. The correct answer was replacement of kinetically favored proteins by thermodynamically favored proteins from a surface over time. So this is Roman effect, where you once have a surface that is exposed to proteins. At the start, proteins which are in bulk quantity or which are in the vicinity of that surface will first adsorb onto the surface, and over time the thermodynamically favored proteins can replace these kinetically favored proteins because they have more stable interaction with the surface. Next question was, when nanoparticles are injected in vivo, the proteins form a layer on the surface of the particles, and this layer, what is this layer called? It's the right answer was protein corona. Next, if cells are transplanted from a human donor to human acceptor, what kind of transplant is it? It's known as allogenic transfer. Next, we had what transplantation in humans. For transplantation in humans, allografting is one of the most common approach. What are the major drawbacks of this approach? The major drawbacks are lack of donors and spread of disease. Next question was, which surface is more favorable for protein adsorption and why? So the right answer was hydrophobic surface, because it is energetically more favorable to displace water and replace with protein, and rough surface because of available surface area. And then lastly, we had this numerical. wherein we were given a straightforward formula and the values were given. We just had to, so the right answer was 8 for this question, and this is the solution. So you'll anyways be getting this slides, and the video will also be uploaded. So if you guys have any doubts, I'll just wait for a while. If not, we can stop this meeting. No? Okay, so shall I end the call?
Thank you, ma'am.
Okay, sure. Thank you.